Hello and welcome to the GenQA Focus On webinar, which today is focusing on the pathogenicity of somatic sequence variants. I'm Professor Sandy Deans and I'm Director of GenQA, and I have great pleasure in being joined today by my colleague Dr. Jenny Fairley, who's Deputy Director and manages the Molecular Pathology EQA assessments run by GenQA. We also have great pleasure in welcoming Dr. Pauline Rehal from the West Midlands Regional Genetics Laboratory and the Central and South Genomic Laboratory Hub in England. So today we're going to has great excitement and really would like to share these findings from a brand new pilot that we have offered in 2020. This has been moving on and developing from our other pathogenicity variant interpretation assessments and really moving more into the somatic field now. And really this has come about through participant demand and our awareness of change in testing strategies in molecular pathology, from moving away from small single gene panels, hotspot targeted testing, into larger NGS gene panels up to 200, up to 500 different genes, whereby we really do need to apply some sort of interpretation on not just the pathogenicity, but the clinical utility of these variants in the cancer patient pathway. Couple of housekeeping issues before we move on. One, please ensure that you're on mute at all times. And we have a live question and answer session after the presentations. So I really would recommend that you pop your question into the box on your screen, and we'll try and get through as many as we possibly can at the end of the presentation slides. Okay, I'm now going to pass you over to Dr. Pauline Rehal to give us a background around somatic variant interpretation and an introduction to the EQA that was provided. So over to you now, Pauline. Thank you.
Thank you, Sandy. Okay, thank you. So I'm going to start off with a background and start talking about variant interpretation. So variant interpretation is about understanding the significance of detected genomic variants in the context of the clinical disease presentation. So we know that interpretation and classification is a very complex area, and many of us have been doing this for a very long time. But in the somatic setting, it's definitely becoming more and more complicated and important. So in the cancer setting, a critical point towards uniform patient management is the assignment of the biological contribution of each variant to the malignancy and its subsequent clinical impact in a specific malignancy. So as Sandy has said very nicely, routine delivery of targeted and comprehensive genomic profiling has resulted in increased detection of novel variants in a continually increasing panel of relevant genes. So it's really important to have clear guidelines to ensure accuracy and consistency, to support data sharing, minimize harm and maximize benefit. Okay, next slide, please. So guidelines. So there are guidelines for assessment of pathogenicity of germline variants, and they're very well established. So these were first in the UK. Some guidelines came out in 2013 together with the Dutch labs, and these have then alluded to the ACMG guidelines that were published in 2015 by Richards et al. And these guidelines are now very well established internationally and used pretty routinely. However, guidelines for assessment of pathogenicity of somatic variants have only been introduced very recently and are still evolving, and I'll talk about guidelines in more detail in a later slide. So what has the main driver been for introduction of these guidelines? It's precision medicine in cancer. So this is about using genomic information to support diagnosis, determine prognosis and optimize treatment plans. And it's about replacement. It's due to the replacement of multiple single tests for well-established targets to single multiplex technologies, namely NGS, but also many others, yielding both known and unknown variants. Next slide, please. Okay, so this is just to give a bit of a recap. So when we're talking about somatic variants, mainly talking about two areas. So gain of function, so this is the type of changes that you primarily see in oncogenes, where they become constitutively active, and what we tend to see in these genes are single nucleotide changes that tend to be missense, copy number changes, and also structural changes. In the other setting, which is loss of function, we tend to see these happening in tumor suppressor genes. So they tend to be in activating type variants. So we see nonsense truncating and intragenic deletions as well. Next slide, please. So somatic variant classification can be broken down into a two-step process. And the first step is when you find a variant is you have to determine what's its pathogenicity. So this is just an example how it would be, how we approach this process within the lab that I work in. So when we see a variant, the type of questions that we ask are, have we seen the variant before? If we have, is it a variant? Was a variant analysis completed within the last year? If it isn't, does the variant have a population frequency above 1%? And then based on this, it follows that basic routes. So once you've done a complete full analysis of the variant and determined whether you need to do a pathogenicity assessment or not, and this will then be taken using in silico analysis tools as well, looking at places like databases. So these are both cancer and germline databases, and they're useful for evaluating somatic variants that have well-studied genomic counterparts, for example, TP53 or BRCA1 or BRCA2. So information on functional studies and other experimental evidence may be available, which could help when collecting evidence towards pathogenicity in a somatic context. So next slide, please. So when the second step, once you've assessed pathogenability, bases are used, looking at literature. So it's important to determine the quality and statistical power of any literature that you review and assess the evidence as a whole. If a biomarker has been recorded in the literature in multiple well-powered studies with a consensus from experts in the field, then that can be used as stronger evidence than literature from low-powered studies where there's only plausible but not confirmed therapeutic evidence. So the levels of evidence are then used to see what the classification could be, and the levels of evidence range anywhere from whether something is an approved therapy or whether there are just one-off case reports or preclinical studies. That's the range that we see. And classification will also be dependent on the context of the clinical question. So is the test being undertaken for diagnostic, prognostic, or therapeutic reasons? Okay. So next slide, please. So one note of caution is about tumor-only testing. So at the moment, the majority of strategies taking place in regard to cancer testing is that most people look only at the tumor and don't always test the blood, which would allow the ability to do a germline subtraction or know what is present in the germline and what is only present within the tumor. So we know now that testing of genes involved in inherited cancer predisposition is now also required for therapeutic management. So a good example of this is, for example, BRCA1 or BRCA2 for PARP inhibitors in prostate cancer and also in ovarian cancer. The problem with this is that it has the potential to uncover clinically relevant information about a patient's germline. So this poses a challenge when reporting back clinically actionable variants in a tumor context, which may be of uncertain significance in a germline context. So for example, missense variants in BRCA1, BRCA2 that are eligible for PARP inhibitor treatment, but what do they actually mean within the inherited setting, and how does this information need to be relayed back? So it was last year or the year before, ESMO Precision Medicine Working Group published some really useful guidelines on how to approach this area. So this table basically brings together what I've described in the previous slide about assessment of pathogenicity and assessment of actionability and how we undertake those. So, and the key point is it is actually quite similar whether it's in a germline or a somatic context. The main difference are the type of resources that would be used, for example, the databases. So specifically for here, disease and gene-specific databases tend to use locker-specific databases in a germline setting, whereas in a somatic setting, it's very much focused on databases such as Cosmic, Bioportal, TCGA, and there are lots of new resources coming online which are very useful as well. So OncoKB and the clinical knowledge base from Jackson Lab as well. Then there are also constitutional bases, databases are used in both settings such as ClinVar, HEMD, CIVIT. Literature searches are important in both areas as well. But again, the focus changes when it comes to looking at functional data. So in the germline setting, it's in the context of pathogenicity, whereas in the somatic setting, it's in the context of actionability. So in a germline setting, we're looking at pathogenicity linked to disease causality, whereas in a somatic setting, you're looking at the impact of that variant on clinical care. Okay, next slide, please. So with regard to somatic variant classification, which guidelines are there? So the ACGS, which is a UK working group, are developing guidelines for consensus approach to interpretation of somatic variants, and this type of work is probably happening in other groups internationally as well. So the main guidelines that are currently in use are the AMP, CAP guidelines, the SCAT guidelines, and also the Belgian COMPEMET expert panel. So I'm going to talk about these in a little more detail in the following slides. There are also the well-established ACMG guidelines and more recently the CANVIG guidelines as well that are also used in the inherited setting as well. So I'm going to talk firstly about the AMP, ASCO and CAP guidelines. So these were published initially in 2017 by Lee et al. So they can be categorized into 4 groups, tier 1, 2, 3 and 4. So tier 1 variants are those that are of strong clinical significance, and they're made up of variants with levels A and B evidence. So level A evidence are biomarkers that predict response or resistance to approved therapies for a specific type of tumour or biomarkers. They're included in biomarkers that are included in professional guidelines that predict response or resistance to therapies for a specific tumour type. Level B evidence tends to be biomarkers that predict response or resistance to therapies for a specific tumour type based on well-powered studies with consensus from experts in the field. And that evidence may come from multiple large studies with supporting outcome data, and tend to see consensus in the literature as well with these types of variants. Tier 2 variants are those that are of potential clinical significance, and they can then be further categorized by level C and D evidence. So level C evidence are those biomarkers that predict response or resistance to approved therapies for a different tumor type, or there can be biomarkers that serve as inclusion criteria for clinical trials, and this may come from multiple small studies with supporting outcome data and functional studies, or approved therapy for different tumor type, tending to be phase 2 or 3 clinical trials with some consensus in the literature. Level D evidence tend to be biomarkers that show plausible therapeutic significance based on preclinical studies, and this may come from functional studies only or isolated studies with preclinical trials, but no absolute consensus within the literature. Tier 3 variants are those of unknown significance, where evidence may be conflicting or it may be absent. This is where there's conflicting literature on pathogenicity or very low-powered studies without outcome data or functional studies. And tier 4 variants are those that are categorized as benign or likely benign, where there is evidence a variant does not have any actionability. So you may have large studies class variants as benign or not stated in the literature at all. Okay, so the SCAT guidelines. So SCAT stands for ESMO Scale for Clinical Actionability of Molecular Targets. So this ranking was determined to support the ESMO Precision Medicine Working Group recommendations for the use of next generation sequencing for patients with metastatic cancers. And there's a lot of similarities with them and the previous guidelines that I've just talked about. So the main differences are that they defined 6 levels of clinical evidence for molecular targets according to the implications for patient management. So the tier 1 variants are targets that are ready for implementation in routine clinical decisions. Tier 2 variants are those investigational targets that likely define a patient population that benefits from a targeted drug, but additional data are needed. So tier 3 clinical benefit previously demonstrated in other tumour types or for similar molecular targets. Tier 4 variants are ones with preclinical evidence of actionability. Tier 5 are variants with evidence supporting co-targeting approaches, and tier X are those variants with lack of evidence for actionability. So some of the tier 2 and 3 have been broken down slightly more, and those are how they've tier 4, 5 tiers for the SCAT. So these guidelines are the Belgian CompEMed. So CompEMed panel stands for Personalised Medicine Commission, and it was created in late 2015, and it is a committee which brings together Belgian scientific expertise within this area. And these guidelines were published in 2019. So this is a 2-level classification workflow system. Variants are first classified into a tumour-independent biological 5-class system. So this is the panel that you can see on the top left-hand side, and subsequently into 4-tier ACMG clinical classification. So the biological variant classification workflow is to define pathogenicity, disease causality. The first step of the biological classification is a technical filtering step to select high-quality variants. Variants that pass the above-mentioned technical filtering steps are then checked for their presence in healthy population databases. The remaining pool of somatic variants then needs to be further classified for their potential tumour pathogenicity. They use what they've called a consensus pathogenic variants list, CPV, which was established for clinically relevant changes in solid and myeloid tumours. So the CPV lists were edited by a consensus of at least 6 NGS experts in solid or myeloid malignancies. So once that's done, it's something needs further assessment. So it's a no clear loss of function mutation variant, it's missense or in-frame deletion. It then goes through to the scoring system, the scoring table. So the scoring table consists of 4 parameters, which you can see under the parameters column heading. So the 4 parameters for which a score between -1 and +2 can be obtained. So the total score will define the class of each variant with a total score of greater than 2, resulting in a likely pathogenic classification, and a score of less than 2 in a variant of unknown significance. So the first parameter of the scoring table is the total number of entries of the specific amino acid change in COSMIC. The second parameter relates to the in silico prediction tools SIFT and mutation taster, and determines if an amino acid substitution is deleterious to a protein function, mainly based on protein conservation with homologous sequences and the severity of the amino acid change. Parameter 3 entails whether the variant has been reported as tumorigenic in functional studies. So looking at things like PubMed, JAK-CKB, etc. Parameter 4 then looks for a detrimental role described for that variant in databases such as CIVIC, ClinVar, OncoKB, and VARSAM. So the sum of the scores of the 4 parameters provides the total score. And these guidelines are also really nice in that they have a very nice section where they discuss about clinical reporting and how to incorporate this information into there as well. Right. Next slide, please. Okay, so I think I'm going to hand over now to Sandy.
Thanks, Pauline, very much indeed. What an incredibly comprehensive summary. So thank you so much for taking the time to outline all the different guidelines and approaches to be used. So just a reminder for you to submit your questions in the live Q&A session, which is after the slide presentation. So please just pop your questions in the box and we'll get to them. Now we will move on to presentation from Jenny, who is going to give us a summary of the results from the EQA for the pathogenicity of somatic sequence variants that was delivered in 2020. So over to you now, Jenny, please.
Thanks, Sandy. So as Sandy mentioned, this EQA was set up because we were aware that this was becoming more commonplace amongst laboratories to perform this type of variant analysis, and also in response to participant demand. So we launched this as a pilot EQA in January of this year. We asked laboratories to submit classification only of 3 variants that we had sent out. So we had 3 case scenarios for these variants. And our aim was to really assess the classification of their pathogenicity in the context of the clinical case scenario we provided. We said that no prior experience or knowledge of the disease or gene was required, because it's, as much as anything, this is looking at the process of variant classification as well as the actual classification itself. And we asked the participants to submit a results pro forma with their actual classifications on it. And we marked based on it was marked on the basis of correct classification of the variant. And as with our other EKs, it's marked by our expert panel, so it's not just one person assessing the results. And we had experts that had already classified the variants for the EQA. So we had 27 registered participants for this EQA from 11 different countries. As we might have expected, there were quite a few from the UK, but as you can see from this graph on this page, we did have representations from quite a number of other countries, which means that we had to think of this as a worldwide approach and not just to UK when we're assessing the reports. Of these registered participants, there were 24 of the labs that actually submitted results for the EQA. I'm going to go over the cases one by one, and I'm going to give an introduction into each case, and then I'll present the results. But Pauline's going to talk to you in the middle about how we came up with the expert classification of these variants. So case 1 was we give the patients mock names and dates of birth. These aren't real, we invent these. So it was a female patient, it was a young female patient who had a histological diagnosis of a tumour which was either an atypical spitzoid tumour or a spitzoid melanoma. And originally this patient had been referred for molecular testing of NTRK to facilitate clinical management, and the tumour content of the sample was 50%. The results were that they didn't actually find an NTRK fusion in this patient. However, they did find an in-frame ROS1 fusion transcript, which was present, which was a PWWPTA ROS1 fusion between exon 1 of PWWPTA2A and exon 36 of ROS1. And our expert classification of this is that it would be a class 2C based on its therapeutic potential, and that you would report this variant. And now I'm going to pass over to Pauline, who's going to explain how we came up with that classification.
Okay, thank you, Jenny. So we have tried to map these against the earlier slide where I discussed how we do pathogenicity assessment and actionability and what steps that we follow. So as Jenny has introduced this case, it was a fusion, and fusions are well-established drivers, but the main thing to check with fusions are whether they are in frame or out of frame, whether it's an active or inactive product. And in this case, we were told that it was an in-frame fusion. So this particular fusion had been reported previously on the COSMIC database. So next slide, please. It was also well established as an approved therapy for a different tumour type. So we know that treatment of ROS1 fusions is approved in non-small cell lung cancer as well. And this was a particular case with this one as well. Next slide, please. So we had multiple lines of evidence towards diagnostic and prognostic status with some consensus for this particular one. So this was the key paper that was quoted for this particular case, and it showed also plausible intervention-based clinical trials. So based on these evidence, we classified it as 2-tier C because it wasn't reported specifically, there wasn't a licensed treatment within this particular tumour type, even though that drug had been used, was licensed. Okay, next slide.
So thanks, Pauline. So this is the results of the laboratories that classified the variants. And from this graph, you can see there was quite a variety of different classifications applied by the different participants of the CQA. The majority of laboratories did provide us with an AMP, ASCO, CAP tier classification. And as you can see, there was some laboratories who would only report it as a tier 1 or a tier 2, and some would actually do the subcategorization of this tiering. When it came to marking of these reports, we actually took a fairly flexible approach to it, because we agreed that although the expert categorization that we had come up with was 2C, that there would be potential reasons why laboratories might apply a tier 1 classification to this variant. I'm going to go into that in the next slide. You can see here there was one laboratory reported it as a tier 3. This laboratory, however, did say they would report the variant, so they did receive a deduction, but it wasn't considered a critical error because they would report the variant. And as actually there isn't approved therapy in this case, we didn't think that constituted a serious clinical implication for it. One laboratory reported that they wouldn't apply a tier to this case because the therapy wasn't appropriate. And as you can see, there are a number of laboratories who also use different classification systems, including the ACMG. 2 laboratories looked at the JAX knowledge base and reported the results from there. And then one laboratory used their in-house classification system. I mentioned to you that the results from case 1 did show a variety of differences, of a wide variation between laboratories reporting either as a tier 1 or a tier 2. The case scenario provided for this case said that it was requested to facilitate clinical management. Now, 5 laboratories classified it as Tier 1 and gave the reason that it was included in guidelines, which is part of the classification system given in Lee et al. In particular, it's included in the WHO classification of skin tumours. However, this is about the diagnostic of whether it be a Spitzoid malignancy or a Spitz-like tumour, and it actually doesn't help discriminate between these different types of Spitzoid tumours, and also doesn't help with the therapeutic options for the patient, which is why we decided to classify it as a Tier 2C rather than a Tier 1.
Mm-hmm.
So case 2, this was a male patient born in 1963 who had a diagnosis of cholangiocarcinoma. They had a multi-gene panel next generation sequencing applied to their tumour. And again, this was to determine management options with a tumour content of 75%. And in this case, there was an IDH1 variant detected, which is an arginine 132 to glycine change. And again, our expert classification of this case was as a 2C. And again, I'm going to pass over to Pauline as to why we classified it as a 2C.
Thanks, Jenny. So with case 2, the variant that was reported was present in a somatic database associated with patient tumour type. So the ARD132 GLY variant is reported frequently in CNS and brain tumours. It's a well-established hotspot change in IDH1. And it's also, you can see, reported in cosmic in bile duct tumours, of which cholangiocarcinoma is a bile duct tumour, relatively frequently as well. And together with that, this variant had a high pathogenicity score that's well established. Next slide, please. So the next step was, it was shown there was a lot of supporting evidence for pathogenicity and also supporting evidence for actionability within this particular tumour type. So plausible therapeutic intervention based on preclinical trials. There were multiple lines of evidence towards diagnostic prognostic status with some consensus, and importantly, the gene was in a tumour type associated cancer pathway. Next slide, please. So these 2 pieces of evidence provide strong evidence for either a Tier 1 or a Tier 2. So inclusion in a clinical trial, phase 2 or 3, was reported in this paper here by Abu and Alpha. And what was interesting is that the drug is not currently approved for use in any tumour types within the EU. So tiering is very dependent on local national guidelines. So for example, in the US, for this particular variant, and for cholangiocarcinoma, the NCCN guidelines do suggest use of IDH1 inhibitor, but however, the inhibitor is not formally approved by FDA. It's only approved in the setting of AML. So, and also what we noticed with this one was that there were different interpretations on literature around the clinical trials, and the strength of the study will also affect the tiering that you apply to this case. So we went with we thought that the study wasn't strong enough to apply a tiering of a higher tiering, so that's why we went for 2C in this case. Next slide, Jenny.
Thanks, Pauline. So as you can see, that the laboratories that participated in the CQA also didn't quite agree on which tiering should be applied. The majority did apply Tier 1 tiering. However, you can see that Tier 2C was the second choice of a lot of laboratories. So there's clearly some controversy as to where this would actually lie within a tiering. It should be noted that we didn't deduct any marks from laboratories. We accepted tier 1 or tier 2 as an agreed answer for this case because of the potential differences as to why you would tier it as a tier 1 or a tier 2. As you can see, there were a few laboratories who also applied other tiering to this case, and no laboratories were deducted any marks for this because we felt that any kind of classification that they had provided was correct. So on to case 3. And case 3 was a patient with prostate cancer, which was diagnosed as stage 4 castration-resistant prostate cancer, and the patients progressed following hormone therapy treatment, and they're asking for molecular testing of this prostate tumour to help determine management. Again, it was a tumour with a high tumour content, about 80%, and in this case, it had a BRCA2 variant detected in it, and the C707G2T, which is an arginine 233C6 lysine potential variant, although as you'll see, that's perhaps less relevant than perhaps other impacts this variant may have. This variant is classed as likely pathogenic according to ACMG guidelines, and also as a tier 1A or a tier 1B. And again, this would be dependent perhaps on the country that the participant was coming from, and as you'll see from Pauline's classification determination.
Thank you. So why was this classified as class 4 likely pathogenic? So this case needed a bit more work to assess the pathogenicity. It was absent from population databases like DBSNP, Nomad. There was no frequency data for this particular variant. Computational analysis in silico tools, 2 of them predicted a splicing defect. Other nucleotide changes have been seen at the same location and shown to induce encoding present in both somatic and associated with patient tumour type and reported as clinically important. And these are the 2 panels that you can see at the bottom, one the literature and the second one from ClinVar. Okay, thank you. Next slide, please. So in terms of actionability, there were multiple lines of evidence towards therapeutic status with some consensus, and multiple large studies with consensus. So namely, PARP inhibitors for metastatic castration-resistant prostate cancer. So there are guidelines of eligibility for this particular drug, and this particular variant would fit into those eligibility guidelines, which is why it was then classified as tier 1. Currently within the UK, it is not NICE approved. There is no approval yet for it at the moment in prostate cancer, only in the ovarian setting, which is why it would be a 1B rather than a 1A. Okay, next slide, please, Jenny.
Thanks, Pauline. And as you can see, the vast majority of laboratories agreed with the tier 1 classification, and that was split between tier 1A and tier 1B. And some of the reasons given why it would be tier 1B, whereas Pauline stated it currently isn't approved by NICE in the UK. One laboratory reported it as a tier 2. Again, we did deduct marks from this laboratory for that particular classification. However, we did not feel it was clinically as severe because they did mention that because class 2 is about eligibility for clinical trials and the like, and we didn't think that was warranted a critical error. However, one laboratory did classify it as a tier 3, and this was because this laboratory classified it as a variant of unknown significance according to ACMG classification. Because we felt that would have potential serious clinical implications for the patient, it was a critical error. There was one laboratory also reported it as a variant unknown significance. This participant only looked in the JAX database, and again, because it is shown to be likely pathogenic, that would have quite a severe impact on the patient potentially, so they were given a critical error. And the third participant was also given a critical error because although they classified it correctly, they said they wouldn't report the variant, and we felt that was very serious implications for the patient. As you can see that when it came to the ACMG classification, there was an almost equal split between pathogenic or likely pathogenic designation. And so this is looking at across all three cases, the AMP, ASCO, CAT classifications of the three cases into tiers. And what you can see from this is a bubble plot. So the size of the bubble, the blue bubble, is dependent on the number of laboratories reporting any particular classification for any case. So for case 1, the biggest bubble was for class 2. I've actually put in class 2A, 2C and 2D together, and class 1A and 1B together in these graphs because there are some people who would only report class 1, 2 or 3, and you can see that clearly for case 1, the biggest bubble is for class 2, and this would be the expected classification of this case. For case 2, actually, there were more people putting this into class 1 and class 2, and in class 3, it's the overwhelming majority. I reported it as class 1. As I mentioned, there's been quite a wide variety of different classification systems used. The majority for all three cases, and the most common classification used is the AMP, ASCO, CAP guidelines. And there were a small number of participants used SCAT guidelines. For cases 1 and 2, there were relatively small numbers using the ACMG guidelines, but that's more used these in case 3, but not surprisingly, because we know that BRCA variants can also be classified according to germline guidelines. There was a couple of laboratories who used the JAC knowledge base, one laboratory who applied in-house guidelines. We had one laboratory who used the Belgian guidelines that Pauline went over at the start, and then a couple that used mainly other databases like OncoKB or Enigma. So overall, there was, as you can see, quite a variability in the classification systems applied and what the actual classification was. It wasn't possible for us to determine some of the reasons for this variation because not all participants submitted enough evidence for us to look at to determine the reasons why. And we also, not all the laboratories took into account the clinical case scenario provided. And we feel that this is probably something that will become more important in the future, as well as classifying the variant is going to depend very much on the context. So the 2021 EQA has already gone live. And in this case, we've asked that laboratories would also submit their evidence for the classification, because it will help us and the other participants to understand this whole process better. And we've also asked in this case for the interpretive comments, which would be included on the reports, because we think this is obviously a key part of the conclusions of this classification. The EQA was distributed to laboratories last week, but it's still possible to enrol because it's open until August the 23rd. And if you're interested in doing so, you can contact us on info@genqa.org. Thank you.
Thank you, Jenny and Pauline, for a fantastic double act. Thank you there. And great to hear that the EQA is now open and it's still an opportunity to participate if you wish. We've got quite a few questions coming in, so if you don't mind, I'll pass them on to you. First of all, there's some queries about whether or not the webinar will be made available or the slides afterwards. So because you've got the registration link, you'll be able to access the presentation and the recording for a month through GoToMeetings, and then the whole filmed recorded session will be made available on the GenQA YouTube account. So if you can't get access or can't find it, then please just drop us an email and we'll point you in the right direction. So first of all, can I start with a couple of questions about reporting and tiering? So first of all, should the tiering be actually stated on the report or not? I was going to come to you, Pauline, first of all.
So I think lots of people don't at the moment. I think what's more important is the overall interpretation in terms of clinical actionability with these. And I suppose it's going to depend on what comes out of the various guidelines and what your local national policies and guidelines will be, as in whether you should report tiering or not. And I think it's also dependent on what the clinician, the referrer, wants to see in the report. So do they want to see a tiering, or do they want text that tells them what the recommendations are?
Yeah, very good point, which ties in very, very nicely to the next two questions, which have come in from different people. So first one, should tier 4 variants be reported? And also, should tier 3 variants be reported? What's your views on that?
So I think tier four are the ones which would be equivalent to SNPs in a germline setting, that there's no evidence of any actionability or pathogenicity. And currently in a germline setting, we wouldn't report these, and I think it would be the same in a somatic setting. If we consider that the panels that we're going to are becoming bigger and bigger, we just won't be able to list everything, and there probably wouldn't be any value in listing everything in a report because the reports are becoming bigger and bigger. Tier 3 might be slightly different, that if you see something with clear pathogenicity but the actionability isn't clear, then you may want to flag this possibly to the clinician, because in future there may be clinical trials associated with it that you may want to go back and reevaluate those over a period.
Okay, thanks, Pauline. Sorry, I think I dropped out there slightly. And that comes on, and you did touch on this, but maybe just to reiterate as well, that how often should variants be reviewed? So you talk about maybe you've seen it in a different patient 6 weeks ago, maybe 6 months ago, maybe a year ago. What's the protocol you have in your lab? Do you just assume that the tiering should be the same, or would you take nothing for granted?
No, so what we tend to do at the moment is, if we've seen a variant before, we usually go back and we review it and see if it's been, how long it's been. So within a year, definitely if it's been approaching a year since we last reported that variant, we would then go back and redo the variant form and see what updated recommendations had been made.
So Jenny, how strict is the assessment going to be for the 2021 EQA? So say, for example, a tier 4 variant is stated on the report, or a tier 3 is or isn't. How are you going to cope with that through the assessment?
I think we'd like to see if, good question, haven't really thought about that until you asked me something. I think it will depend on people's local guidelines whether they do that reporting. So because we've asked them for interpretive comments, I think we should have a better idea of what their reporting is. And then we've always asked that people, if they have specific guidelines that they perform in their local laboratories, that they should let us know, because we can take that into account with marking as well.
Sure. Okay, thank you, Jenny. And it'll also, I guess, depend the clinical context and also absolutely. Yeah, the actual, and I guess a change that you mentioned as well is that we'll be asking for the evidence used for the classifications. So again, I think we'll have to be a bit flexible on the assessing and deductions based on what evidence was used or potentially what evidence was missed.
Yeah, agreed.
So,
And we understand that this is quite a new process for laboratories, which many of them are still implementing. So
absolutely.
we have tried to be as flexible as we can with this.
Right, Pauline, can I come back to you? A very specific one about case 3. Did you apply PS3?
Oh,
You're probably going to have to go back to your notes for case 3.
Yeah, I will have to go back to my notes if you bear with me.
Right. So we'll come back when you're doing a review. We've got a statement here which is perfectly correct, that in the UK, due to the EMA licensing of drugs, that the private patients and also in the devolved nations are not under NICE approval process, so the drugs can be potentially given to those patients despite no NICE approval in place, and also the fact that this EQA is global, and we've seen that we've got participants from many different countries, that we do take into account national policies on approved drug approvals, so we wouldn't deduct in those cases for any labs. So thank you for raising that.
Yeah, I think I'd like to reiterate that we have tried to be flexible with this. So although we've come up with what we said was our expert tiering, we didn't deduct if you didn't entirely agree with it, depending on the circumstances.
Yeah,
because we do appreciate there are differences in different countries, and that is quite important to take into account.
Thanks, Jenny. So Jenny, can I stay with you for another EQA-based question? Are there examples of good reports available for new participants? And this is something we get asked all the time right across all of our EQAs. Have you any updates for that, please?
Not for this EQA, because we didn't ask laboratories to submit the reports. We only asked them to submit data. But we've looked at doing that for other EQAs, and we'll definitely consider doing that for some of our other EQAs again.
And what we've started to do actually is, instead of giving you a template or an actual full report, because we're very aware that labs are really constricted by the LIMS you have in place, whether you've got integrated reporting or not in place, and therefore to start saying that you should have it in a table format or in bold or a particular layout is something that often labs cannot do. So we would rather go with nice terminology and phrases, how to get the message across clearly without misinterpretation. So perhaps that's something, Jenny, as we go forward with this new pilot, that we can incorporate that for our participants.
Absolutely, and we can give it good examples of interpretive comments in this case, hopefully, for the next EQA that we're doing. So.
Thanks, Jenny. Pauline, have you gone back to your case 3 notes? Can we come back to you about PS3?
Yeah, I can't tell you if we applied PS3 locally because I can't see those results. But what I can say is that what was applied was PM2, PP3 and PP5 PS4. So those were the ones.
Right.
So I can go back and review.
Thank you, that'd be interesting. And also somebody else has put in the question box about case 3, is it appropriate to do case counting to apply PS4 stroke PM5 moderate, as BRCA2 prostate cancer is not rare?
Probably yes, I think it is appropriate to do that. Yeah.
Okay. Thank you very much. Okay, going to databases now. We have a question, which is quite a long one, but I'll read it out for you. So it's in reference to using COSMIC database to classify a variant based on frequency of that variant in different cancers. If the mutation is not present in too many samples, then it will be undermarked based on Belgian guidelines. COSMIC is only collating published data, so many mutations found in the clinical setting are not getting published or reported in a public database, which is something that we're all very aware of. Yeah. So how much, therefore, should one rely on the frequency of mutations detected in the COSMIC database? So it's basically saying what, in your view, is the quality of the data in COSMIC?
I think what we tend to do is we tend to look across different databases, so things like OncoKB and CKB, and look at those as well and bring all that information together, that rather than relying on one, because as they've just pointed out, there's different values to different databases. So I wouldn't rely completely on one, because we know this, we have the same situation with all of them in terms of curation and how much data has been submitted.
So is that also equivalent to ClinVar? Because we know ClinVar's put a lot of time and effort into enhancing the quality of the data in their database with their, their star rating and expert panel review. So would you use that over and above databases? Would you only use ClinVar that's been reviewed by experts?
No, I think you would want to look at the different databases and get a consensus. That's the way, probably the best way to work, I would say.
Okay, great, thank you for that. I totally agree. So we've got one here, but can you address the use of PP5 given recommendations to remove the ACMG AMP reputable source criteria for the interpretation of sequence variants? Do you want to address that one, Pauline, or do you want to do that in a written response to give you time to check?
I think we'll do that in a written response, if that's okay.
That's Heather, do you want to just pop your email address in the questions if you wish? If not, you can email info@genqa.org and we'll come back to you. Hopefully that's fine. Also, there's also another very specific question around BRCA2 variants of comprising of deletions of exon 1 in the upstream UTR. So perhaps again, Magdalena, could you let us know your email address and we'll come back to that, because it's a very specific question and I don't want to put Pauline and Jenny on the spot whereby they may be quoted at some point. Thank you very much. And I think brilliant. Thank you, Magdalena. I think that's pretty much all our questions coming in today, but I think that's been great because it's covered the EQA element, it's covered the databases and also specific requirements of how to look at their actual pathogenicity of the variants themselves as well as in the clinical setting. So thank you very much, everyone, for your questions. Jenny, I'll just come over to you now for some final comments, please.
So we'd very much welcome your feedback on the webinar. Those of you that have attended in live in person today will get a survey sent to you following the webinar. The webinar will be available for a month on the GoToWebinar system for those people who've registered for it. And then in a month's time, it will also be available on our GenQA YouTube channel, which you can subscribe to. And those of you who participated live on the day will get a participation certificate sent out to you from GoToWebinar. Unfortunately, we can't give participation certificates to those people who haven't actually attended the live seminar due to the way that this is run. Carrying on with our variant classification, the next focus on topic is going to also be around classification of variants, in this case postnatal copy number variant classification, and that'll be towards the end of June. We look forward to seeing you then.
Thank you, Jenny, very much. So just finally, a big thank you to you for joining us today, and also to our expert panel, that's Pauline Rehal and Jenny Fairley. So I hope you found this a useful webinar. Thank you. Bye-bye.